Bismillahirrahmanirrahim. I'm Dr. Shazia Kul. Today is my second class with you, that is lab guide on layers of retina. आप लोगों को आवाज आ रही है? जी, आप लोगों को आवाज आ रही है? जी, today is our class on lab guide on layers of retina. आप लोगों को मेरी आवाज आ रही है?
Yes, madam.
Okay, let's start. Please mute. So today I will tell you on the layers of retina and what are the basic structures that form histologically 10 layers in retina. So in this diagram, you are able to see on one side, there is the image and histological image, and on the other side, the schematic diagram of the retina. So by comparing these two diagrams, you can easily understand the basic structure of layers of retina. We will start from this side, that is outer side, and the first layer is layer of pigment epithelium. You can easily see in schematic diagram that layer of pigment epithelium is towards outer side. So where is outer side? You should know that outer side is towards the choroid plexus and inner side is towards the vitreous body. So outer side pigment epithelium is basically a low cuboidal epithelium that is resting on the basement membrane. This is basement membrane of the pigment epithelium and this is low cuboidal epithelium. So this epithelium has a rounded nucleus in the basal region, and in the apical region you can see brownish black pigment, that is the melanin pigment. That's why this epithelium is called pigment epithelium. And apical cytoplasmic projections are also visible. So these are projecting into the layer of rods and cones. So this is schematic. On this side, this is the layer of retinal pigment epithelium, and this basement membrane of the retinal pigment epithelium is tightly bound to the choroid layer to form the Brooks membrane, and cytoplasmic processes are projecting towards the layers of rods and cone. Here you can easily appreciate the nuclei in the basal region and brownish black pigments in the apical region. So this is the first layer. Now come towards the second layer. Second layer is the layer of rods and cones. So in this layer there are processes of the rods and cones. So what are rods and cones? These are basically photoreceptor neurons. Rods are responsible for dark vision in dark, and cones are responsible for the color vision. So there are three basic colors, that are green, red and blue. So the processes of rods and cones, they form the layer of rods and cones here, and these are the processes of rods and cones. So this central area is basically the cell bodies of the rods and cones. This is the cell body of the rod that are shown in the color, and the rod shaped cones are by the side. So in the third layer here, this layer is outer limiting membrane. So outer limiting membrane is basically this area in the schematic diagram, this area. So outer limiting membrane is a thin membrane that is formed by the junction between the apical process of this large molar cell and the bodies of the rods and cones. So here there is junction between the bodies of the rods and cones and the apical area of the molar cell. This large cell by the side is, this whole cell is molar cell. It is extending from this layer towards the end, inner side. So this is a very large cell, molar cell. It is basically a supporting cell, and outer limiting membrane is formed by these two adhesions. So next comes the fourth layer, that is outer nuclear layer, this one. In histological image, this outer nuclear layer, can easily appreciate the nuclei. So these nuclei are basically nuclei of these rods and cones. So these nuclei of rods and cones are arranged in this outer nuclear layer. So in the outer part, they are especially nuclei of the cones are arranged, and in the inner side, nuclei of the rods are arranged. Please keep this thing in mind that cones are relatively less in number and rods are more in number. Next comes the outer plexiform layer. This outer plexiform layer is basically an area of synapses between the processes of the rods and cones and the processes of the various types of cells that are present in the next coming inner nuclear layer. So here you can see the synapses. This is the area of synapses. Here the processes of the rods and cones are making an anastomosis with the various cells, different types of cells that are present in the next coming inner nuclear layer. So next is inner nuclear layer. Here you can see that in inner nuclear layer the cells of the various sizes are present, and relatively it is covering a large area because these are the nuclei of the different types of cells that are present in this layer. So these different cells, the cells are shown in this schematic diagram. You can see bipolar cells, these amacrine cells, by the side Muller cell, and this one horizontal cell. So these four types of cells: horizontal, bipolar, amacrine, and Muller cells. Their cell bodies of all these four types of cells are present in the nuclear layer, inner nuclear layer. So next comes the inner plexiform layer. In inner plexiform layer, again it is a synapsis between the processes of the inner nuclear layer and dendrites of the adjacent next coming ganglion cell layer. So this is this area in the schematic diagram. I can show you that this is a synapsis between the dendrites of ganglionic cells and processes of these all four types of cells. So this is inner plexiform layer. Next comes the ganglion cell layer. So in this layer you can appreciate one thing, that the nuclei are relatively of larger size. So these are nuclei are basically the nuclei of very large ganglion cells, and they have large nucleus. These ganglion cells are third order neurons, and they are basically multipolar neurons. So their dendrites are making synapses with the inner plexiform layer, and their axons are exciting to form nerve fiber layer in the next coming inner layer. So next comes the ninth layer, is the nerve fiber layer. This nerve fiber layer here in the image, this is basically made up of these axons of the optic nerve fiber, or the axons of the ganglionic cells that are forming the nerve fiber layer, and these nerve fiber layer is exiting in the as a optic nerve from the retina and going towards the visual cortex. So innermost layer is the inner limiting membrane. This inner limiting membrane is basically the basement membrane of these Muller cells. So this is the Muller cell. This basement membrane of the Muller cell forms the inner limiting membrane. Now you can very well appreciate the Muller cell. On one side it is forming the inner limiting membrane, then its cell body lies in the inner nuclear layer, and its apical projections form adhesions when the bodies of the rods and cones form the outer limiting membrane. So this is the basic structure of histological structure of the retina. So this is again image of the layers of retina. So you can appreciate various layers again, starting from outer side. This is first layer is pigment layer, and the second is the layer of rods and cones, then outer limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglionic cell layer, nerve fiber layer, and inner limiting membrane. Now next, now we will study the retinal layers, their anatomy, histological anatomy in detail. So retinal pigmented epithelium is actually not a part of neural retina but intimately associated with it. So layers of rods of cones contains the outer and inner segments of photoreceptors. So what are photoreceptors? Photoreceptors are basically rods and cones, and they have processes which are arranged in the form of outer and inner segments, and these outer and inner segments are separated from each other by small constriction. Outer limiting membrane is the apical boundary of the molar supporting cells, as I already showed you in the diagram. Then next comes the outer nuclear layer. It contains the cell bodies of the retina and rods and cones. Outer plexiform layer contains the processes of retinal rods and cones and horizontal and bipolar cells that connect to them. Inner nuclear layer contains the cell bodies of horizontal, amacrine, bipolar, and molar cells. So what are these cells? Basically, these are the neurons. Horizontal amacrine are the association neurons, bipolar are the direct conductive neurons, and molar are supporting cells. Inner plexiform layer contains that is the seventh layer. It contains the processes of these type amacrine, bipolar, and ganglionic cells that connect to each other. Next comes the eighth layer, that is the ganglion cell layer containing the cell bodies of the large ganglion cells and their large sized nuclei. Ninth layer is layer of optic nerve fibers. It contains processes of ganglion cells that lead from the retina to the brain, that is the visual cortex. Inner limiting membrane composed of basal lamina of the molar cells, as I showed you in the diagram. So next comes the non-visual photoreceptors. So basically, what are the non-visual photoreceptors? As I showed you that in the ganglion cell layer, a subset of ganglion cell in ganglion cell layer contains a chemical that is 11-cis retinal that is bound to melanopsin, which serves to detect quality and quantity of light during each 24 hours drawn in the cycle. Now, in this slide, I am going to tell you that I have told you about the different layers of retina, but we should know that in these different layers, what are the basic tissues that are present? Retina has basically two basic parts, a neural part and a non-neural part. Neural part is again made up of photosensitive part and non-photosensitive part. So in photosensitive part, there include rods and cones, and in non-photosensitive part, there are different types of cells that I will tell you in next coming slides. And the pigmented part, that is a non-neural part, it is made up of layer of pigment epithelium. Layers of retina, 10 layers of cells and their processes, they constitute basically retina. So now I will exactly tell you the different types of cells that are present in the retina. There are neurons and supporting cells. So number one, photoreceptors. What are photoreceptors? They are basically neurons that are photosensitive, and these are rods and cones. Number two are the direct conducting neurons. Direct conducting neurons are the bipolar cells and ganglion cells. To keep this thing in mind, that rods and cones are basically first order neurons, and the bipolar cells are second order neurons, and ganglionic cells are third order neurons. From ganglionic cells, axons emit and they form the optic nerve and finally take the nerve impulse to the visual cortex where image is formed. So number three is the association neurons. These are horizontal, interplexiform and amacrine neurons. They are not direct conducting, but they conduct different types of cells with each other. Supporting cells are the molar cells. Molar cell is a very large cell that I have already told you in detail, that is present and forms a part of retina. So various types of neuroglia are also present because retina is an extension of central nervous system. So these neuroglia are made up of microglia and astrocytes. As you know that microglia are basically part of mononuclear phagocytic system. Astrocytes are other cells with processes and they have many supporting roles. This is again a diagram. On one side histological hand drawing, on the other side there is schematic diagram just to show you again the structure of retina. You can well appreciate the different layers of retina here. This is pigment epithelium, then the layer of rods and cones, external limiting membrane, then outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, and then this ganglion cell layer, fiber layer, and the limiting membrane. So one thing that you will especially appreciate in this diagram that you can see that I have made this red color structure just to show the capillary plexus in this layer and this layer. So what is it? Basically, retina is a neural structure and very important structure for vision. So its nutrition is also very important. Outer one third of the retina, that is the pigment side, outer one third of the retina, it is supplied by the diffusion from the choroid capillaries, and this area does not have blood vessels, but in inner two-third it is supplied by the branches of the central artery of the retina, and this central artery of the retina gives various branches that form capillary plexus in the especially in the inner nuclear layer and the ganglionic cell layer. So these plexus we can see in the section. So in the schematic diagram, you can see the shape of rods and cones. So these cones, their segments are cone shaped and rods are rod shaped. And moreover, you can see the large sized ganglionic cells, their larger nuclei, and their shorter dendrites that are forming synapses with the inner layers and their long axons that are exiting to form the optic nerve. So these are the, again, a repetition. So this is again, these are the layers of retina. Now, when you see in the lab, in your exams, you have to identify the slide and you have to write down the two identification points. So the identification points are written on this side. So the most important identification point is that when you see the slide of retina, you can easily appreciate that the retinal structure is arranged in the form of various layers, and if you meticulously see, you can well appreciate the 10 layers. And number two is the presence of rods and cones. So rods and cones is next to pigment epithelium on the outer side, there is a layer of rods and cones, and then next coming there are cell bodies or the nuclei of the rods and cones in the outer nuclear layer. So these are the identification points of the retina. So this is a microscopic image of the retina. This is exactly what you would see in your lab under the microscope, light microscope. So one thing you can first you will see that there are retina is arranged in the form of different layers, and then when you start concentrating, you can well appreciate the arrangement of these layers. So this is basically pigment epithelium. After the pigment epithelium comes the layer of rods and cones, then next coming layers. So you can see that there is a gap between the pigment epithelium and layer of rod and cone. So this is basically, as I already told you, that pigment epithelium is not tightly bound. It's loosely attached to the next coming layer of rods and cones. But so that's why during preparation of slide, there is sometimes due to shrinkage and during processing, there is a gap that becomes visible on the histological slide. So this is because of processing. This is the drawing, that is the how you will draw the different layers of retina in your notebook. So exactly you will draw like this. So you can easily see that this is the first pigment epithelium. You have to draw the outer cell membrane with the eosine pencil, and on the basal side you have to make its basophilic nucleus and brownish black, in the apical region brownish black melanin pigment. Then like this you have to draw different other layers, and you will also draw plexiform capillary plexus in these inner nuclear layer and ganglionic cell layer, as I have shown you in this. And you will a thin membrane draw thin membrane here in the form of inner limiting membrane and outer limiting membrane and nuclear in nuclear layers. So this is exactly how you draw the layers of retina. This is the Now in this video, I will tell you how to draw retina. So please watch it. So outer plexiform layer is basically synapse. Now I will tell you how to draw different layers of the retina. First layer is layer of pigment epithelium. It lies on the basement membrane. Pigment epithelium is low columnar cells with basal nuclei and apical brownish black pigment. Second layer is layer of rods and cones. Layer of rods and cones is basically they are processes of rods and cones. Third layer is outer limiting membrane. Outer limiting membrane is junctional complex between the processes of the Muller cells and the cells of the layers of rods and cones. In next section, I will tell you what are the Muller cells and how they form the outer limiting membrane. Fourth layer is outer nuclear layer. Outer nuclear layer is basically it contains the cell bodies of the rods and cones. Fifth layer is outer plexiform layer. So outer plexiform layer is basically synapses between the outer plexiform layer is basically synapses between the processes of the this second layer and the dendrites of the next coming six layers. Now sixth layer. Sixth layer is inner nuclear layer. Inner nuclear layer basically contains cell bodies of four different types of cells. So you will draw the nuclei of different sizes. So these four different types of cells are horizontal cells, amacrine cells, bipolar cells, and Muller cells. So as I told you, that cell bodies of the Muller cells lie in this inner nuclear layer. Seventh layer is inner plexiform layer. Inner plexiform layer is again a synapses between the process of the sixth layer and next coming eighth layer. So what is eighth layer? Eighth layer is basically layer of ganglionic cells. So it contains nuclei of cell bodies of the ganglionic cells. You will draw relatively larger nuclei in this layer. So this is ganglionic cell layer. Ninth layer is layer of nerve fibers. These are basically axons of 10th layer is inner limiting membrane. So inner limiting membrane is basically basement membrane of the Muller cells. So Muller cells have basement membrane here, cell bodies in the inner nuclear layer and processes in this outer limiting membrane. So we will draw one another thing. We will draw capillary plexus in sixth layer and in this layer, ganglionic cell layer. So outer 1/3 of the retina is supplied by the capillary plexus present in the choroid, and inner 2/3 is supplied by the branches of the retina. They form plexus in these two layers. So these are the how you will draw 10 layers of retina. Thank you. Any question, please? क्वेश्चन आप सब समझ आ गए? क्वेश्चन है तो प्लीज पूछें आप लोग. Okay. So today we have covered lab guide on layers of retina, and next time inshallah we'll meet soon. Allah Hafiz. You will draw all these layers of retina in your notebook and send me your assignment.